Hey everyone, welcome to Study Side. Today we are going to study mitotic cell cycle. So let's get started. Cell cycle refer to the series of events that take place in a cell, resulting in the duplication of DNA and division of cytoplasm in organelles to produce two daughter cells, which means it is a way in which a single cell organism divide to form two daughter cells. Now the question arises, why a cell divide? Basically, when a cell grows, there arises a little disbalance in a ratio of its nucleus and cytoplasm. This disbalance can affect the normal functioning in which is why cell divides. So, the answer to the question why cell divide is to restore its nucleocytoplasmic ratio. Let's discuss some terminologies for cell division. Division of cytoplasm is known as cytokinesis and division of nucleus is known as karyokinesis. Things to know before we start the main topic. A human cell divides in 24 hours and a yeast cell divides in 90 minutes. Cell cycle is consist of many events, so we will try to study it step by step. The major event of cell cycle are interphase, M phase and G naught phase. The interphase lasts for more than 95% of cell cycle. This phase is also called resting phase because it's time during which the cell is preparing for division by undergoing both cell growth and DNA replication. Interphase is further divided into three phases: G1 phase or gap 1, S phase or synthesis, G2 phase or gap 2. G1 phase is interval between mitosis and DNA replication. During this phase, the cell is metabolically active but do not participate in replication of DNA. S phase is interval between gap 1 and gap 2. Here, the amount of DNA doubles with no increase in chromosome number. During this phase, the centriole also replicate in cytoplasm of the cell. G2 phase or gap 2 is the interval between DNA replication and M phase. Here, the cell synthesis protein and prepare for cell division. Now comes the most interesting phase of cell cycle, the G0 phase or called quiescent stage. The cells that do not divide further exit G1 phase to enter an inactive state. The cell no longer divide unless called on to do so, but it remains metabolically active. In animals, mitotic cell division is only seen in diploid cells. There are some exceptions where mitosis is seen in haploid cell. Example, male honeybee. A quick recap. In G1 phase, cell is metabolically active. In S phase, the DNA replicates. In G2 phase, protein synthesis occurs. In G0 phase, where cell exit G1 phase. Before we start M phase, let's discuss about chromosome. Chromosome has two symmetrical structures called chromatid or sister chromatids. Each chromosome has a constriction by which sister chromatids attach, and this region is called centromere. Spindle fiber attach at the centromere during cell division. On the side of centromere, a disc-shaped structure is present called kinetochore. The M phase is the most dramatic period of cell cycle. This is also known as equational division as number of chromosomes in the progeny are same as parents. M phase is divided into prophase, metaphase, anaphase and telophase. Prophase is the first stage of mitotic division. The centriole which has undergone duplication during S phase of interphase now begin to move towards opposite poles of the cell. Chromosomal material condensed to form compact mitotic chromosome. Chromosomes are seen to be composed of two chromatids attached together at the centromere. Centromere, which has undergone duplication during interphase, has begun to move towards opposite pole of the cell. Each centromere radiate out microtubule called aster. The two aster come together with a spindle fiber forming mitotic apparatus. It simply means chromosomes are condensed in this phase. The mitotic spindle also starts to form the microtubule and proteinaceous components. The disappearance of cell organelles like Golgi complex, endoplasmic reticulum, nucleolus also occurs in this stage. The next phase is metaphase. By this stage, condensation of chromosome is completed and they can be observed clearly under the microscope. This is the stage at which morphology of chromosome is most easily studied. Spindle fiber attached to kinetochores of chromosome. Chromosomes are moved to spindle equator and get aligned along the metaphase plate through the spindle fiber to both poles. Here, the chromosome align at equator with the help of spindle fiber, and this alignment in the equatorial region is known as metaphasic plate. Anaphase, the centromeres plate and chromosomes separate. Chromosomes move to opposite pole. It's simply as each chromosome moves away from the equatorial plate, the centromere of each chromosome is towards the pole and hence at the leading edge, with the arms of the chromatids trailing behind. Now that the chromosome reached their respective poles, they started to decondense and tend to collect in a mass in two poles. Chromosome cluster at the opposite spindle poles and their identity is lost as discrete elements. Nuclear envelope assemble around the chromosome cluster. Nucleolus, Golgi complex and endoplasmic reticulum reform. Telophase is known as reverse prophase, as all the events that happen in prophase happen once again in a reverse form in telophase. Now that the karyokinesis is done, it's time for the cytokinesis. This process is accomplished differently in animal and plant cell. In animals, this is achieved by appearance of a furrow in plasma membranes. The furrow gradually deepens and ultimately joins in the center, dividing the cell cytoplasm into two. In plants, valve formation starts in the center of cell and grows outwards to meet the existing lateral valves. The formation of new cell wall begins with the formation of a simple precursor called cell plate that represent the middle lamina between the walls of two adjacent cells. In some organisms, karyokinesis is not followed by cytokinesis, as a result of which multinucleated condition arise, leading to the formation of syncytium. Significance of mitosis. The growth of multicellular organism is due to mitosis. Cell growth results in distributing the ratio between the nucleus and the cytoplasm. A very significant contribution of mitosis is cell repair. Thank you everyone. That's it for today. Let's meet next time.